Okay, so now we're getting into a little bit more of the metabolism. And so let's just briefly recap. We have, what is this again? All right, glycolysis. Okay, so to start it off, you have to prime the pump and actually use two ATP molecules to push phase one to get all the way to glyceraldehyde 3-phosphate. And then once you get into that, you start making ATP in phase two or stage two. And so by the time stage two is done, you are ending up with pyruvate. And that's usually what they tend to talk about in animal phys, human phys. But we're going to also mention stage three, which is getting into the fermentation products. And depending on what microbe you are, what enzymes you've evolved, you will actually have different, and what resources you have around you, you will have different stage three pathways. So they're just showing three here. But a key thing to look at is you start with one glucose and you take it all the way into stage three. You don't see glucose all the way broken down to CO2. It's an incomplete breakdown. The lactate, that's not CO2. Ethanol is definitely not CO2. Okay, so what this is implying is that if you don't break glucose down all the way to CO2, you don't get as much ATP made in the process. Now, pyruvate. So if you're a microbe and you're a savvy microbe and you have some nice options available to you, if you run glycolysis and come to pyruvate, you're going to assess your environment. Say, okay, I made it to pyruvate. Do I have any oxygen around me? And if you're the type of microbe that can use oxygen, when you get to pyruvate, you're not going to push it into stage three fermentation products. That would not be wise. All right. But that's only if you're a microbe that is happy to use oxygen and do that. So if you get down to pyruvate, I kind of think of it as a branch point. It's at that point the microbe's going to stop and go, do I have oxygen? And if so, then the pyruvate will get taken over into the Krebs cycle or the TCA cycle or the citric acid cycle. How do you learn it in biochem classes? What do they call it? Do they? And your book calls it the TCA cycle. So tricarboxylic acid, hence the TCA acronym, because it's difficult to say that over and over. So here is the citric acid cycle, TCA cycle, Krebs cycle. You see pyruvate coming in and running this stage next now. And so it is a cycle. So you have to think about pyruvate, and then you get the conversion to acetyl-CoA. You get some CO2 taken off at that point. And then it's acetyl-CoA and oxaloacetate that will react and get citrate and CoA. And then that's the start of the rest of the cycle then. And again, you know, we're not going for step by step. What I want you to pick out from this is a couple of things. What's one good thing happening here? Pardon? Reducing power, is that what you said? Yeah, reducing power, NADH or NADPH, depending on the microbe. And then FADH2, those are reducing agents, so that's really good. What's another thing? GTP, yep, or ATP, depending on the microbe. So this is substrate level phosphorylation. They don't really show it here, but there is a little blip of a phosphate getting attached, actually the succinyl, and then the enzyme takes it and runs it, and you get your GTP in that little reaction. Okay, so that's really good. And then the third thing that's really good is you see the CO2. It's getting, so your glucose that became pyruvate is now getting broken down to CO2. That's good. That means that you're getting some good bang for your buck. As you completely break down the glucose, you're getting some nice reducing agents from that, some ATP or GTP. All right, so I think we were right about here when we last met. So, by the way, I think I redid these and posted them, updated the slides yesterday in EduCat. But anyways, I think we left off here, roughly, thinking about this idea that when most people hear the phrase oxidative phosphorylation, they immediately assume oxygen's involved. And that's fair when you are talking about animals and humans and, you know, other animals, and yeah, okay. But really, oxidative phosphorylation in this class, when you think about microbes, it should be thought more like, oh, it's an electron transport lane phosphorylation event. That's what we got going on here. And in this context, oxidative, when you hear that phrase, it's really the idea that electrons are being lost or stripped off of a molecule. You're oxidizing, all right? So oxidative phosphorylation is trying to imply that you have, in this case, it's going to be this electron transport chain process where electrons are given off by a donor. So there's the oxidative, and then accepted by a recipient or acceptor, all right? And then you have this chain reaction of the electrons getting passed down the chain. And in that process, you get phosphorylation to occur. So please, on the next exam, I ask about oxidative, don't tell me oxygen. This is really a big concept in microbiology, that you think about oxidative just means we're stripping electrons off of something in this case. Keep it at that. Yes.
What other terminal acceptors?
We're gonna get into that. Oxygen, if you are a savvy microbe, you'll pick oxygen. If oxygen's available, that's your number one. But some microbes, many microbes don't have that availability, so then they have to put something else at the end of the electron transport chain. And it can be all kinds of different things. Nitrate is a popular one. You'll look at that in lab. So we'll talk a little bit about that. So yes, we do aerobic respiration. You're doing that right now, where oxygen is the final or terminal electron acceptor in your mitochondria. If you use oxygen, you get higher ATP yields by far than if you just run fermentation. So if pyruvate got pushed into stage 3 of glycolysis, that's way less ATP. So this idea of take pyruvate and put it into the TCA cycle or citric acid cycle, and then get those products over to the electron transport chain with oxygen. That's your best bang for your buck. So in a human, we're doing this in our mitochondria. But microbes do it in mitochondria. Where do they do it? So this is an electron transport chain process, donor to acceptor. Yeah. Cell membrane. They pack it all into their cell membrane. And so again, you know, remember mitochondria were once ancient free-living microbes. So microbes don't have the mitochondria, but they certainly have adapted and put everything into their cell membrane instead. So many microbes use something other than oxygen. So especially if oxygen is something that is either unavailable or maybe it's toxic to them, then they'll have to choose something else besides oxygen. But if a microbe can use oxygen, they will certainly get that aspect of the electron transport chain running where oxygen can be the terminal electron acceptor. And what's happening is you'll get this transfer of electrons down the chain, so to speak, and oxygen will be at the very end. And as that's happening, it creates a situation that can allow for ATP synthesis. So what are we talking about here? Here is an example of a microbe's electron transport chain. So you can see that's the cell membrane that they're showing. And then you see certain complexes, 1, 2, 3, and 4. Okay. So what you are seeing here is these complexes are players in the game for the electron transport. The Q cycle as well, the cytochrome C. So these are all players in the game to pass electrons from donor to acceptor on down the line, so to speak, until you finally get to the terminal electron acceptor, which in this case is oxygen. Okay. So now you're seeing what's happening with the
citric acid cycle or TCA cycle, you see here NADH coming in and FADH2.
So that's from the TCA cycle. Those products were made. So they get shunted over to the electron transport chain and they're like, hey, I'm a reducing agent. That means I have electrons I can donate off. And so they donate them off into this transport chain process at slightly different points, but they're very happy, chemically speaking, to give off those electrons and pass them on down the chain until oxygen gets those electrons. And once oxygen collects enough of those electrons, it gets converted, and protons, it'll get converted to water. But that's not what the microbe's going for. Their goal is not to make water. Their goal is to create a situation where they have a little bit of free energy to pump protons across the membrane. That's their goal. That's what they're really doing here. And so this is pretty universal. I mean, microbes came up with it first a couple billion years ago. But you know, when you think about how we do it in our mitochondria, well, they were once ancient microbes. So this is a pretty universal thing. And what you see is
this EO that you see on the left here, all of this graphed out here, this EOB.
And this is, I'm gonna be talking about this in a little bit here. But you see these numbers, negative 0.22, and then a 0 mark, and then some positive values here. So this is a kind of a reducing potential scale. And I'm gonna get into this, but basically anything that's a negative number. So all of like complex 1, all these players in the game, they are, what that's really saying is they're really happy to give off electrons. They do that quite nicely. And then as you get past the 0 point and into the plus values, those players in the game are more happy to accept. And it creates this situation where you get this little bit of free energy as you go down, so to speak, this reducing potential. And basically it just allows for an easier time of passing the electrons. And in that process, it creates a little bit of extra energy, free energy that can allow for pumping across the membrane of the protons. And so whenever you think about electron transport chain processes, you should always marry it to the development of a proton gradient. That's really the goal here. That's what the microbes are like. I'm just trying to create a situation where I have a little bit of free energy on the way here, like key spots where I can pump some protons. We're not going for oxygen turning into water. That's really not it. So if you get a proton gradient across a membrane, what's one thing you can think about? What will that do for the what could it do for the microbe? You get a nice proton gradient. What could the microbe do with that? Yes.
Protons move into ATP. And then ATP can be made.
Absolutely. So this artist who drew this, I mean, this is fine, but they didn't finish the thought. And I see this a lot in medical, or not medical, but science illustrators, I should say, where they leave off the key thoughts because they're not necessarily biologists. They're told to make an electron transport chain drawing. But what you need to think about is they should have included ATP synthase in that drawing because that's finishing the thought correctly. All right. So if you get that proton gradient and the protons are charged, so they can't, that gradient wants to dissipate and it wants to diffuse. Those protons want to diffuse, chemically speaking, across the membrane, but they can't go through that hydrophobic, they're charged. So they go through the ATP synthase. And as they do that, it actually creates a little bit of a torque force. And you see here how ADP and inorganic phosphate react to get you the ATP. So I wish the artist had put that in. Hey. Here. But they didn't. So you have to link these ideas together that proton gradient really is a source of potential energy and it can be used to create ATP
if they flow through the ATP synthase. So depending on what class you're taking or what book you're reading, you might hear the proton gradient expressed as an electrochemical potential across the membrane, or you might hear also known as a proton motive force, also known as a proton gradient. I tend to say proton gradient. It's shorter and easier. Plus, it very succinctly states what's getting passed across the membrane. It's the proton. So don't forget, I wish they would have drawn the ATP synthase down here so that they could have shown the protons coming through and the ATP being formed. Pardon?
Like not in the cytoplasm.
Yes, this is all the way out of the cell, and then they come back in. Other questions on the electron transport?
What they come back in?
Through the ATP synthase. So it's not being shown here, but they have to travel through the ATP synthase. They call the ATP synthase a little molecular motor because it actually, these subunits actually rotate. And that allows the physical pairing up. It allows for ADP and inorganic phosphate to get close enough to actually react. So it's the cause of as the protons are flowing through, it's causing that torque force. Pretty amazing. So with the electron transport chain, you don't need to know the agonizing detail of step by step. But you should be able to describe it adequately, that you understand it, there's many donors and acceptors on down the line of electron transfer going on. And that the key thing is they're linking it to development of a proton gradient. Obviously, knowing that NADH and FADH2 are the players that are really helping with, here's some electrons, here's some protons. We can pump those protons across, the electrons feed into the chain process. So now you can imagine how these are linking together. You have glycolysis going to pyruvate. The microbe looks around, do I have oxygen? Yep, okay, I'm going to send it to the citric acid cycle, get my NADH, my FADH2, a little bit of energy, get NADH and FADH2 over to the electron transport chain. And when they do that, they get a nice proton gradient. So that allows for some nice ATP. And when you look at it, here's another representation of what a microbe might be doing with their electron transport chain. And this time the artist included the ATP synthase, really big one. So but that's okay. I mean, it's important that you recognize this is the goal. It's to make that ATP. And so this process, yes, it's called oxidative phosphorylation, but not necessarily because oxygen is there, because microbes can take that out and put something else there. And it's still called oxidative phosphorylation, even without oxygen. So just be careful about that. And they don't have a mitochondria, they put everything in their cell membrane. So if you link all of this together, glycolysis, the citric acid cycle or TCA cycle, and then the electron transport chain system. And what I like this one who was trying to show how they all fit together. I remember when I used to learn all this, I had to learn everything in isolation. I never was really taught how they all flow together so nicely.
Do they call it oxidative phosphorylation because NADH and FADH2 lose electrons, they get oxidized?
That's part of it. Yeah, that's part of the reason why. I mean, basically oxidative, they're trying to get across the point that you have stripping of electrons and feeding into the electron transport chain. Yeah, you got it. Okay. So here you can see, they're trying to show the payoff in terms of ATP. So that's glycolysis. When that's done, you get 2 ATP with pyruvate, or no, here, the TCA cycle. You get a little bit of ATP, and then you get those lovely reducing agents that then feed in to the electron transport chain, where you have oxidative phosphorylation here. Oh, here they're saying, yep, if you have oxygen at the end, what you end up with is like 34 ATP by running the electron transport chain. So you can see if you are a microbe that has oxygen and you can use it, you do. Because this gives you the best payoff for ATP by far. As far as we know, oxygen is the best electron acceptor on the face of the earth. There's no better chemical out there, molecule out there. Oxygen is the best in terms of living systems using it to do their goal of making ATP. You get more ATP using oxygen at the end of the electron transport chain. So if you're a savvy microbe and you can use oxygen, you will. But there's going to be plenty of times, like E. coli right now is in your gut. Trillions of them are in your gut, and they all like to use oxygen. But there's little microenvironments in your gut where all the microbes in that area might be scarfing up all the oxygen. And so that area goes anaerobic for a while. And then if you're lucky, you'd be something like E. coli, where you go, okay, I don't have oxygen. I'm going to switch to a different metabolic pathway. That's okay. I can survive. And so E. coli is what we call a facultative anaerobe, which means if oxygen is present, it will use it. If oxygen is not present, it'll just switch to something else and it'll survive. And so facultative anaerobes are a really nice microbe to beat because you definitely have a lot of options. So questions on these three cycles? How they blend into each other, flow into each other? By the way, how many people are in genetics right now? My office isn't very far from the genetics lab. I have fruit flies visiting me regularly. Are you working with fruit flies? So I just telling my other class this morning, I do box breathing, you know, have you heard of box breathing? Where you, if you're like stressing out about something, you take a nice slow breath in for 4 seconds, hold for 4 seconds, breathe out for 4 seconds. I did that in my office and a fruit fly flew up. I just saw a little, what I thought was a fruit fly here. That's what made me think of it. Ever have a fruit fly go up your nose? It's not fun. Ugh. So yeah, be careful with no fruit flies if you do box breathing. So, but anyways. Okay, so breathing, microbes, E. coli loves oxygen if it's available, yes. So it's the biggest payoff for ATP. Now, microbes might decide that they're going to use that proton gradient for something else besides ATP, at least for a little while. And so we already talked about the idea of there's those proton gradients that get established in order to get the flagella to rotate. And so they'll use a proton gradient to get their flagella to rotate around and be modal. So that's a really good option. If you're modal, you might have to devote some of that proton gradient to that idea. But you're seeing some other ideas here too. Uptake of nutrients. Sometimes they have to pick out some protons in order to bring in certain nutrients. And then sometimes if they're getting exposed to toxins like antibiotics, some microbes become very good at the antibiotic comes into their cell and they just efflux it out. Say nope, and they have the proton gradient energy to say nope, I'm getting rid of it. Okay, and so sometimes that's a trick that they'll use, which is not good for us, but good for them. All right, so yes, the proton gradient is awesome for ATP, but just realize there's other things they can do with it in order to stay alive. So that proton gradient is really universal and it has many uses. Okay, I'm going to shift into the next slide set. Is that posted in Educat now? It didn't get posted? I meant to post it. Huh? Yes, every once in a blue moon. I'm going to show you that actually. It's bizarre. How do you know that? Yeah, yeah.
I don't know about that one. I know sodium. Eh. Seriously, I didn't. Okay, let me just look at this really quick. All right, let me post the right one. Okay, it should be there now. It's called part 1. In the sense that I'm past the intro section now. Okay. So, let's go to here. Okay. All right, so now what I'd like to do is dive in a little bit more to some of these processes that the microbes are doing. Okay. So now we have to think a little bit more about what microbes are capable of. And you could spend a whole year just talking about microbial physiology. And I took 2 semesters of this in grad school and it was still not enough. So obviously I'm giving you the highlights, but we need to think about redox reactions and this idea that during redox reactions, you have these electrons getting transferred. And again, if you strip electrons off of something, it's in its oxidized state. And if electron is getting accepted, it becomes reduced. That's what they're showing here. Now, when I was first learning redox reactions, they say it's electrons getting transferred. And yet it was really hard for me to see that until someone explained to me, well, the electrons are coming in the form of the hydrogen. And so it's often shown as the transfer of hydrogen. Because if you remember from basic chemistry, each hydrogen contains an electron and a proton. And so it is a transfer of electrons. It's just hidden away, so to speak, in hydrogens getting attached to the molecule. Okay. So keep that in mind as you're working through this. Redox, so the RED stands for reduction, OX for oxidation. So that is a redox reaction that you just saw there. And so why would a microbe do this? It's because it creates a situation where they can get that proton gradient. And that will then lead to ATP production if they need it. So technically, we say they occur in pair, these reactions, they occur in pairs, 2 half reactions. And so you have something that's going to donate the electron and one that's going to accept the electron. So once the electron gets donated off, then that molecule has become oxidized. It's lost an electron, so it's in its oxidized state. If a molecule accepts an electron, then it's in its reduced state. Now, there's a couple of different acronyms to remember how this works. Did you learn any of those in other chem classes? What's one? Oil rig and Leo the lion says GER. Is there any others? Those are the 2 I've learned. I don't know of any others, but they both work. And it's really good. I use oil rig because it's shorter, but loss of electrons is oxidation, gain of electrons is reduction. And oil rig, oxidation is loss, reduction is gain. Okay. So those are good acronyms to keep in mind as you're trying to keep straight what's going on in the process. Now, what you're seeing here are 2 half reactions up top. You see hydrogen, and they're breaking it down into, yes, with H2, you have 2 electrons, 2 protons. They're saying hydrogen is going to be the one donating.
And the reducing potential, E sub zero is the prime mark, that's the reducing potential. So how good is it at donating electrons? Chemically speaking, how happy is it? Do they donate really happy, or is it grudgingly going to give off electrons? Any negative value, so this is negative 0.421, that's a really good reducing potential. So what that's saying is H2, hydrogen gas, is really happy to give off electrons, very happy to do so. Very obliging in that regard. Now here, oxygen, on the other hand, oxygen has a positive reducing potential, which means it does not give off electrons. You have to force that. Instead, it's very good at accepting. I tend to think about oxygen as like the Hulk. You know, and if the oxygen doesn't get its electrons, it smashes. Because if oxygen doesn't get enough electrons, it starts creating free radicals, kinds of issues, and smashes your proteins and cell membranes and your DNA. So oxygen, I always imagine as the Hulk sitting at the end of the electron transfer chain. Give me electrons or I'm going to smash. All right, and it just loves taking electrons. And that's what makes it the best electron acceptor that we know of on the face of the planet. So we know that certain molecules, they just vary. How happy are they to give off electrons? Maybe they don't want to give them off at all, like oxygen. They vary. And you can put them on this, what we call a redox tower, to kind of show their place in the scheme of whether they like to give up or accept electrons. So that E sub zero, we'll call it the reducing potential or reduction potential. And so the way this works, if it's a nice negative number, fairly large negative number, which believe it or not, that is up top there, that's saying that they're very happy to give off electrons. Whereas the positive value, like with oxygen, it's an excellent electron acceptor, but it hates to give off electrons. So I know some of you have had redox concepts in other classes, so hopefully this is ringing some bells. Now, I think your book has this table. I do not like this table for some reason. My brain just does not click with it. So I'm going to use this one. I think this one works better to conceptualize what's going on. So this is called a redox tower. And you can see here that they're written in a certain way on the tower. So what we have happening here, if you look up here, where they have the 2H plus / H2. So I've written that here. Okay. So the way they have this written, by convention, all the redox couples that go together, they always show it with the oxidized form placed to the left of the slab. So 2H plus is the oxidized form, and then H2 is the reduced form of hydrogen. Okay. So that's how you interpret this. And then you can see that there's a reducing potential scale on here, the E sub zero prime. And it's, you know, all the way from negative 0.6 on this one that they've shown to all the way down to like plus 0.9. Okay, so what that's saying, and then look at your zero mark. Your zero mark is right here. Anything above the zero mark are molecules that are pretty happy to give off electrons. The further up you go on the tower, the happier they are, the more capable they are of giving off electrons. Once you hit the zero mark and go into positive numbers, now the reverse happens. Those are the molecules that are good at accepting. And the further down you go, the better and better they are at accepting. Make sense so far? Okay, so your H plus is your oxidized, and the H2 is the reduced form of hydrogen. So let's look at a couple of examples. They have them outlined here by different colored arrows. So the rule is that they have 3 arrows here. So let's look at the black one first. Number 1, they show
they all start with the H plus / H2, but they go to different other molecules. So the way to look at this, if you're trying to figure out like what would get you more ATP, it's basically the further the electron falls on this tower, the more bang for your buck. So that black line, you can see the electron doesn't fall very far. It goes from this couple up here just down to fluoride succinate. So that's a pretty shallow jump for the electron. But if you have the electron going a little bit further, so the red line number 2 down to the nitrate, it fell further. So the way to interpret that is you're going to get more energy, bang for your buck, and more ATP should be coming from this. Your third line, the blue line, you can see it goes all the way down to O2 water. That's the best situation. The electron falls really far in the furthest in these 3 examples. That means you're going to get more bang for your buck. So when you see redox towers, oxygen's always at the end because oxygen is the best electron acceptor that organisms use. So if you're a microbe, what you're doing is you're assessing your environment for these types of molecules. And you're like, where do they fit on the redox tower? I want a lot of bang for my buck. And if I have oxygen, I'm going for that. But, I don't have oxygen. Well, then maybe you do better, do I have iron? That would be good, because it's just a little bit up from oxygen on the tower. So that would get you still a lot. And then you see the nitrogen here, the nitrate and by nitrogen. That would be another really good option. But if I only have like fumarate or succinate, if I have something like that around me, yeah, okay, it works, but it's not as good. All right, so what microbes are doing is not only do they have to constantly assess what's around them in terms of molecules and nutrients, but then they have to, grab the ones that are the best for the payoff. And oxygen would win in every case. So let's look at this. So if we say that H2 is the reduced form of hydrogen, and you look up there, so basically anything above the 0 mark, whatever's to the right of the slash, that's what's going to donate off electrons. Okay, so of this pair that I've written here, H plus H2 is H2. That is actually going to donate off electrons in these 3 reaction pathways that they've outlined here. Okay, so this is the one. And then let's look at the number 1 idea. So basically what you're saying, you would write, okay, H2, and then, so H2 gives off electrons, and then when you look down here, it would give off electrons to the oxidized form of that couple. What's the oxidized form of that couple? Is it fumarate or succinate? Fumarate. So what you would say then, okay, you go H2 plus fumarate, those are the substrates, and then that yields, that would be your arrow, succinate, and you have to show water instead of protons forming, but that's okay. So that's kind of the game that you're playing. Above the 0, the donors are going to be to the right of the slash. Below the 0, the electron is going to get given to the member to the left of the slash, because you have to do a reduction oxidation. Donate electron, accept electron. Anything that's oxidized goes below the 0. All the oxidized partners will be the ones accepting. Yes.
We have like a question or we'll have to maybe go off this.
Yeah, that would be highly likely. So let's look at what they have written out here. So in all cases, it's the H plus / H2. Well, we know above the 0, what's going to donate off electrons is to the right of the slash. So H2 is going to donate off electrons, and then the arrow goes to fumarate succinate. It must be giving it to the oxidized version, so that would be fumarate. So H2 donates off the electrons off fumarate, and that yields succinate. And you can see when they calculate the thermodynamics of that, the free energy is negative 86. That's good. Why? Because basically it takes negative 32 kilojoules per mole to make ATP. So the microbe's game is, I want to create a situation where electrons are donated and accepted in such a manner that I get a more negative number than negative 32. So negative 86 is more negative, so this is good. You can get a couple of ATPs out of this. So it's a game to the microbe. It's like, I want some nice negative kilojoules per mole energy so that I can make ATP. I know I gotta get to at least negative 32. If I go beyond that, negative 86, okay, I got my ATP. Then you can see as a comparison to the number 2 chemical equation where H2 gives off the electron to NO3 minus. NO3 minus is the oxidized one, it's to the left of the slab. And then you're left with nitrite, water, and you got negative 163. That's even better. So again, you're seeing the further the electrons fall, the more bang for your buck. You get more free energy to make ATP, even more ATP. And then the last one, all the way down to oxygen, you can see a nice payoff, negative 237 kilojoules per mole. You can make many ATP off of that. So they're using these redox reactions. Basically, it's just a game to, I want to create some free energy so I can make some ATP. Yes,
I'm going to find out.
that's free energy. Just know it's free energy available to do something. So let's do a Google form. Let's see if we can work through this while we're all together. Whoops. Ooh. Try this. You can look at your notes, you can talk to each other, but see if you can work through these questions. We almost done? Okay, let's quickly go over. Okay, so which redox couple pair will be donating electrons? A or B? A, very good. Why? More negative, it has a reducing potential, it's negative, so that's the pair that's going to be donating. So which molecule would actually donate the electrons? Acetate. Acetate's the reduced version, and so acetate will be donating off electrons. And then which molecule will actually receive the electrons? Yes, Fe3+, the ferric form of iron. So, okay, I was looking at the graph coming in real time and it looked like most of you were getting it. So, but we will recap this again on Monday to spend a little bit of time getting this organized in your head. We'll go over it again on Monday, and then we got to get more into fermentation ideas as well.